Hello everyone, I'm Deepak Modi from the ICMR's National Institute for Research on Women's Health, and the topic for today's discussion is spermatogenesis and sperm production. The male reproductive system consists of the testis, where spermatogenesis occurs, and it is also the site for the male hormone production. Epididymis is where sperm maturation happens. Vas deferens is where the sperm gets transported, and the seminal vesicles and the prostate produce secretions which are required for sperm survival. Today's presentation is focused on the testis, which is an oval oblong-shaped structure, which is covered by the epididymis in which sperm maturation occurs, which is connected to the vas deferens. Now, if we cut a section through this testis, you would see the outer covering of the testis, which is the scrotum, is made up of the tunica. Inside to it are long coiled tubules called as the seminiferous tubules, and these seminiferous tubules connect to form the retitestis, which then connect to the epididymis. If we cut a section through this, and when we see it here, this is a testis, this is a section of a human testis, we can see these multiple seminiferous tubules which are looking round like this, and inside it are the cells which are doing the spermatogenesis. Now, what are the cell types in the testis? The sperm tubule has two compartments, a basal compartment and the luminal compartment. From basal compartment to the luminal compartment, you can see the different kinds of cells. There are the Leydig cells, which are outside the testis, outside the seminiferous tubules. Then there are blood vessels, there are fibroblast cells, and there are immune cells here. The layer which is protecting or making up the seminiferous tubule is called as a peritubular myoid cells, and inside it are the Sertoli cells, which are also called as the nerve cells of the testis, and they are these spermatogonias and where the spermatogenesis process is happening. So the spermatogenesis process requires, as I said, the spermatogonia, which are the primordial cells, which are the primary cells, which continue the process of spermatogenesis towards production of the mature sperm. So the mature sperm are derived from the spermatogonia, but the spermatogonia is actually derived from a primordial germ cells. It is interesting that the primordial germ cells do not develop into the testis when the gonad is developing, but rather it is derived from outside. And here is a diagram which explains it. The gonad develops as a thickening next to the mesonephros, which is the developing kidney. This thickening has all the other somatic cells except the germ cells. During the course of development, very early, around 6 to 8 weeks in humans, the primordial germ cells are developed from the base of the allantois, and they migrate inside the developing embryo. Remember, there are two testicles, so there are two genital ridges which are formed, and on each of the genital ridges, the germ cells go and migrate and they colonize there. And look at this beautiful movie which is showing it. You can see these germ cells coming as a stream from the hindgut, and they're going on either sides and colonizing the genital ridges, and this is how the testis is formed initially. Now remember, here right now everything is a mixture of cells. You can see this genital ridge here. The green colored are the Sertoli cells, and the cells inside this are the germ cells. During the course of development, between 7 to 10 weeks of gestation in humans, these seminiferous tubules are formed by the Sertoli cells, which are going to form round structures, and they form an oblong tube. And then they, inside these tubes, are entrapped the germ cells. And you can see this formation of tubulogenesis. This is obviously in the mouse because it's not possible to do these experiments in the humans, but you can see these beautiful tubules which are being formed during the course of testicular development. And inside each of these tubules are the germ cells. The outside cells are the Leydig cells, and this is where the fetal testis is formed. Now remember that germ cells do not mature to form the sperm way until puberty. So if we cut a section through a prepubertal testis, you will see these tubules inside which there are Sertoli cells, and there are these spermatogonial cells which remain quiescent. Upon the induction of puberty, in the adulthood, the spermatogonial cells, in response to various hormones, then initiate spermatogenesis. And the key hormones which are required for induction of puberty and continuation of spermatogenesis are kisspeptin, GnRH, that is gonadotropin releasing hormone. Then you have the follicle stimulating hormone, the luteinizing hormone, testosterone. Studies have also shown that estrogen and progesterone in small amounts are essential for spermatogenesis, and there is inhibin for negative feedback inhibition. And the details of this we will see in this slide, where the kisspeptin, which is produced by the hypothalamus cells, stimulate the production of GnRH in the hypothalamus. The GnRH then goes to the anterior pituitary, and the cells of the anterior pituitary makes two hormones, follicle stimulating hormone or FSH, a luteinizing hormone called as the LH. This hormone gets secreted, and in the testes, they act on the Leydig cells, and they stimulate the production of testosterone by the Leydig cells. The testosterone is then released into the body, and it is required for secondary sexual development during puberty, and during the normal course also it is required for the normal libido. The testosterone locally produced by the Leydig cells also diffuse inside the seminiferous tubules and act on the Sertoli cells and instruct the Sertoli cells to now initiate spermatogenesis. Parallelly, the follicle stimulating hormone which is produced only acts on the Sertoli cells, and then it produces a lot of goodies which allow the process of spermatogenesis to continue. It also produces an androgen binding protein which binds to testosterone and giving the testosterone to these cells in very small amounts. Now, this entire process has to be very tightly regulated, and there is something called as a negative feedback inhibition. Now, as I told you that LH acts on the Leydig cells to produce testosterone. As the amount of testosterone builds in, it impacts the hypothalamus and tells the GnRH to get suppressed. And then the GnRH suppresses, the FSH levels and the LH levels reduce. Similarly, the FSH, which is acting on the Sertoli cells, produce inhibin, and inhibin now blocks the production of FSH, and therefore the amount of FSH also reduces. So testosterone and inhibin together act as a negative feedback. As the gonadotropin levels, the FSH and LH levels reduce, there is a reduction now locally in the production of inhibin and testosterone. The negative feedback inhibition goes off, and then the new cycle of production begins. And therefore, it is not surprising that the levels of these hormones during the entire day in the body keeps fluctuating according to the diurnal rhythm. And this measurements of these hormones together tell us how well the entire HPG axis or the hypothalamo-pituitary gonadal axis is functioning. So in infertile men, many a time we measure testosterone levels to get a judgment of how good are the Leydig cells and their functions on the spermatogenesis, because it's a very crucial hormone required for spermatogenesis. We also measure FSH and LH levels to understand how well the pituitary is functioning. And if these levels are low, there could be a problem in spermatogenesis. Many people do also SHBG and inhibin measurements, but they do not help much clinically. But now you would understand for sure that bodybuilders who take a lot of androgens and testosterone often suffer from infertility simply because it suppresses the GnRH, and the GnRH levels are low, and therefore it doesn't produce much of FSH, and the cycle is blocked. So excessive testosterone is not good for the health, and excessive gonadotropins will lead to again overproduction of testosterone, which will create a problem with this entire process. So this is how the endocrine regulation of spermatogenesis is done. So to summarize, the germ cells are located inside the seminiferous tubules. The Sertoli cells are also located inside the seminiferous tubules. However, the Leydig cells are outside. The germ cells respond to various products from the Sertoli cells. The germ cells respond to the Sertoli cells respond to FSH, and the Leydig cells respond to the hormone LH. Germ cells produce a number of molecules whose identities are not known, but they do impact the process of spermatogenesis. Sertoli cells mainly produce inhibin and SHBG to give the negative feedback to FSH, and SHBG to control testosterone release, and LH helps in production of testosterone from the Leydig cells. The functions of germ cells is to undergo meiosis and produce mature sperm. Sertoli cells are the nurse cells required for differentiation of the mature sperm and also make the BTB, which I'll tell you a little later. And Leydig cells make testosterone, which is absolutely essential for spermatogenesis. Clinically, conditions like obesity actually impacts the pituitary and inhibits the production of FSH and LH and thereby suppressing spermatogenesis, leading to infertility. Number of toxins can impact the Sertoli cells and the germ cells, thereby compromising sperm quality. Smoking and nicotine products from the smoking can affect Sertoli cells and Leydig cell function directly and therefore compromising spermatogenesis. Now, what does the actual process of spermatogenesis looks like? As I told you, there are spermatogonial cells which can undergo mitotic division and replace itself, or they can exit from mitosis and go into the spermatogonium fate, which are diploid, and these form primary spermatocytes. The primary spermatocytes undergoes the first meiosis division, which is a reduction division, and this undergoes a second meiotic division, which is a second reduction division, to finally produce the mature sperm. The first meiosis division is where the DNA crossover happens. Remember, we get one set of chromosomes from our mothers, the other set of chromosomes from our fathers. These are very different DNAs. All the DNA characteristics are crossed over during this meiosis stage 1 through leptotin, zygotin, pachytin, and diplotin stages. And this then allows the crossover DNA. The meiosis 2 now segregates into different sperm. So meiosis reduces the chromosome number from 46 to 23. So each sperm now carries haploid 23 numbers of chromosomes. And it is interesting that one spermatogonium, therefore, can lead to essentially 4 haploid sperm cells. Now remember, this entire process of spermatogenesis is very sensitive to chemotherapy and radiations, and therefore cancer patients, many a times when they experience chemotherapy, they lead to infertility. Now, there's some interesting facts of spermatogenesis. This entire process from spermatogonia to formation of mature sperm is a long drawn process, and it takes about 74 days. That means 1.5 months before the process has begun, and the final product takes about 2.5 months it takes to make the mature sperm. However, the amount that it is making, the total amount of spermatogonial cells and the rate at which it is doing is very, very high. So it's like a factory, and it is astonishing to know that it produces about 1,000 sperms a second, or about 150 to 300 million sperms every day. So it's a huge factory-like condition. And the process of spermatogenesis allows a genetic shuffling, which I've already told you. And the process of spermatogenesis also requires this blood-testis barrier. Now, what is this blood-testis barrier? The Sertoli cells form a very tight junction with each other and does not allow the exposure of the blood to the rest of the cells in the testis. Now, why this is important? Remember, I told you that when the cells are developing, there are only Sertoli cells and the spermatogonia cells. Now, the immune system is developing when we are before puberty, and therefore the immune system thinks that the Leydig cells, the myoid cells, the Sertoli cells, and the spermatogonial cells are self, and they should not be rejected. However, spermatogenesis is initiated after puberty, and the learning of the immune system is already completed. Now, these cells can actually become targets of the immune system, and autoimmunity can be generated, and these cells can be killed. So to prevent that, the BTB is there. The blood-testis barrier keeps these cells in 2 separate compartments, and therefore preventing these cells to autoimmune attack. At the same time, the BTB also creates separations of the microenvironment within the germ cells, so that different sets of germ cells can get different kinds of goodies as and when and where they need. And finally, this BTB is also excellent toxin inducers. We may inject a lot of toxins, but the testis is prevented from this damage through this BTB. However, under number of conditions, the blood-testis barrier can be damaged. For example, heat, heavy metals, pesticides, they all impact the blood-testis barrier, and they can expose the blood testis barrier to the immune system gets exposed because of the damage, and there could be autoimmune infertility. Also, low amounts of FSH and testosterone or nicotine can impact the blood testis barrier, thereby compromising spermatogenesis. Now, the testes, remember, are outside the body, and they keep the temperature of the testis slightly lower than the body temperature. Our body temperature is 37 degrees, but spermatogenesis is not normally progress at 37, and it requires 34 to 35 degrees. So the scrotal muscles actually pull out or keep the testis out or in, depending on the body temperature. It can regulate, and therefore it can continue the sperm production so that the temperature is well maintained below the 37 degrees. And therefore, men would understand this, that at some point the testes go inside, at some points really the testes hang out, and that is actually the body's response to a temperature control, so that the testis always remains at a lower temperature, and the spermatogenesis can progress. But in conditions like cryptorchidism, where the testis fails to descend, there could be many reasons for it, and such testis is exposed to higher intra-abdominal temperatures, which can impair germ cell development and spermatogenesis, and if left uncorrected, it can lead to reduced fertility or infertility. And therefore, in patients with infertility, it's important to ask history of undescended testis. Transient fever, now you understand why, can reduce the sperm count, and therefore history of fever in patients with low sperm counts is very essential. Now, once the spermatogenesis has reached, the process of spermatogenesis actually completes at a round spermatid stage. The next set is a cytodifferentiation of the round spermatid to the elongating sperm. It occurs in 4 stages, and this entire business is called as spermiogenesis. And this spermiogenesis involves elongation of the round spermatids, formation of the flagella, removal of the extracytoplasm, which is phagocytosed by the Sertoli cells, and release of the mature sperm. Very important change which occurs in spermiogenesis is the histone to protamine modification. Usually in all our cells, the DNA is bound about proteins called as histones. But this is a very loose packaging. In the sperm, the DNA has to be very, very tightly packaged, and this packaging is allowed because these histones are now removed, and another set of proteins called as protamines come in place. And this is called as histone to protamine transition, where 2 additional proteins, transition protein 1 and transition protein 2, take over between the elongating and the condensing state. And in the final mature sperm, all the histones are replaced by protamines, and small subsets remain behind. Now, when there is DNA packaging defect because of the abnormal spermatogenesis, things are affecting the process of spermatogenesis. If the packaging has gone defective, it can lead to infertility. Such a defective packaged DNA is at risk to oxidative stress, and therefore it can lead to DNA damage, and all of this can have infertility and adverse pregnancy outcomes. Again, I must remind you, environmental toxins like endocrine disruptors and smoking increases oxidative damage, and this can lead to DNA damage, which can be a cause of infertility. Now, once the mature sperm are formed, they go inside the structure called as epididymis, which is nothing but a long coiled tubule, but here no spermatogenesis happen. The mature sperm are coated by secretions from the epididymis and undergo morphological modifications, and they go a process called as maturation. Remember, the testicular sperm are immature, they're not capable of motility, and they're not capable of natural fertilization. Only when they transit through the epididymis, there a number of modifications make these sperm highly motile and capable of fertilizing the egg, both in vivo and during in vitro fertilization. But in infertile men, this has been bypassed. Many infertile men most of the times do not do spermatogenesis. But if you look at the testis very carefully, there'll be small areas where spermatogenesis is escaped, and these patches can be then aspirated. So what one does is they do a process called as testicular biopsy or testicular sperm extraction, where they poke into the testis and try to remove a lot of cells. And then embryologists actually look for these immature sperm. Round spermatids are immature sperm, and they inject them into the egg. You can see this process of ICSI here, where the egg is held by a pipette. The immature sperm is taken in and it is directly injected. There is no need of motility. There is no need of maturation. And this injection now gets into the egg. This is a haploid spermatid, gets into the egg, they fuse, and a lovely embryo is formed. So essentially now, even if you have immature non-motile sperm, which happens in many cases of infertility, fertility options can be given. Biological parenthood can be given by doing ICSI followed by embryo transfer. Live births have been reported. They're very healthy humans. So this is one of the ways by which infertility, because of issues with spermatogenesis, can be overcome, giving these patients a hope for biological parenthood. So to summarize, spermatogenesis occurs in seminiferous tubules of the testis and it's initiated at puberty. Spermatogonia undergo mitotic division, they undergo meiosis and spermiogenesis to produce mature haploid sperm. And this is a very continuous process. The mature, and it is under the hypothalamo-pituitary gonadal axis, mainly the hormones FSH, LH, and testosterone are the three important key hormones for regulation of spermatogenesis. And the mature sperm acquire motility and fertilizing capacity only when they transit through epididymis, and defects at any of these stages can lead to problems with sperm production and maturation and therefore male infertility. But techniques like ICSI can overcome any of these barriers, giving a hope for fertility in many of the infertile couples. Thank you, and I'll be happy to take questions.